The strength of antigen-antibody interactions depends upon affinity, avidity, and cross-reactivity of antibodies. Let us discuss them one by one. Affinity is defined as the strength of a reaction between a single antigenic determinant and a single combining site on the antibody. In other words, it is the strength of reaction or interaction between an epitope of an antigen and a paratope of an antibody. So this type of reaction strength involves the sum of all the attractive and repulsive forces. So an antibody which has low affinity to an antigen binds to it weakly and dissociates readily. However, an antibody which has high affinity for an antigen binds to it tightly and remains bounded for a longer period of time. Generally, the better fit of an epitope, as in this case with the antibodies of the paratope, result in a more favorable interaction and result in high affinity. As you can see in the second case, uh, the epitope doesn't fit properly into the paratope of the antibody, hence results in a poor interaction and low affinity. Affinity is calculated as a equilibrium constant that describes the antigen-antibody reaction, here as you can see. So most of the antibodies have high affinity for an antigen, and the affinity of the antibody for an antigen is one of the most important factors in determining the antibody efficacy in vivo. The affinity at one binding site does not always reflect the true strength of an antigen-antibody interaction. When a complex antigen containing multiple repeating antigenic determinants or epitopes are mixed with antibodies containing multiple binding sites or paratopes, the interaction of antigen with the antibody at one site will increase the probability of reaction at second site. So in this case, affinity alone would not be able to uh, give us any clue about the strength of the interaction. So in that case, another term is used which is called avidity, which determines the overall strength of binding of an antigen which has many epitopes and has an interaction with the multivalent antibodies. So avidity is influenced both by the valency of the antibody as well as the antigen. So avidity depends upon the affinity of the antibody for the epitope as well as on the valency of both antigen and epitope. So here you can see when one epitope is involved, we calculate the affinity. When more than one epitope is involved, we calculate the avidity, and the avidity increases with the number of valency of the uh, antibody as well as that of an antigen. So sometimes high avidity can compensate for low affinity. For example, secretory pentamer of IgM often has a lower affinity than IgG, but the high avidity of IgM, because of its higher valency, enables it to bind to antigen effectively. Although antigen-antibody interactions are highly specific, but in some cases, the antibodies generated against one antigen can cross-react with unrelated antigen. This is called cross-reactivity. In cross-reactivity, an individual antibody combining sites can react with more than one epitope. As you can here see that one antibody can interact with multiple different types of antigen. This is called multi-specificity. This results when two different antigens share an identical epitope, or in some cases, the antibodies bind to epitopes which are unrelated but have a similar chemical properties. But in this case, the affinity for cross-reacting epitope is lesser than that of the original epitope. An example of cross-reactivity is that the antisera or the antibodies generated against human insulin can react with the insulin of a pig or sheep. So students, it's time for our first concept checkpoint. So you have to answer these two questions. In the first question, you have to write down at least two differences between affinity and avidity and the significance of each of them to study the immunological responses. And the second question is you have to find out how viral or bacterial antigens can result in cross reactivity and give two examples. Try to find out their answers and we will discuss them in our interactive session. Antigen-antibody interactions result in elimination, destruction, or neutralization of the pathogen by various different mechanisms like opsonization and phagocytosis, complement fixation, and lysis of the cell by membrane attacking complex, by activating natural killer cells using antibody dependent cellular cytotoxicity, by preventing the adherence of bacteria to the host cell, by neutralizing the viruses, neutralization of toxin, and activation of allergic response by sensitization of mast cells. Now let's look how antigen-antibody interactions leads to opsonization and enhanced phagocytosis. The first step is like the antibody binds to the antigen on the surface of the bacterium to form an antigen-antibody complex. This complex binds to the surface of the macrophages and the neutrophils by means of their Fc segment. These cells, these phagocytic cells, have Fc receptors on the surface to which the Fc portion of the antibody binds. As a result, the whole bacterium, which is coated with the antibody, becomes attached to the surface of the macrophages. These macrophages then surrounds the bacteria and endocytosis. Within the endocytic vesicle, lysozyme fuse with it and result in phagolysosome. Within the phagolysosome, the bacteria is digested, and in opsonization, three main antibodies are involved: IgG, IgM, and IgA, which results in opsonization.